Hi class, this is Jeff. Today's lab is on the streak plate. It's on page 16 of your lab manual. And we're going to teach you this technique. It's a method of separating bacteria from its neighbors. Okay, so let's take for example, you took a nasal swab from a patient, and in your nose there's going to be all sorts of different microorganisms. And so in order to do tests on them to identify which organism you are actually interested in, you have to have them separated. And so when they're separated in a test tube or on a petri dish plate, then it's called a pure culture. Okay, and so we have to do that before a test would be valid. Because if it's still a mixture and you test it, well, you don't know whether the organism that you want is giving you the test results, or is it the neighbor that was in there when it wasn't supposed to be, and it's giving you a test result. So there's total uncertainty, and it just wipes out your test usefulness. So you have to have them in pure culture. Okay, and so when we do a streak plate, we're going to use our inoculating loop, which we have here, and we have a TS agar plate or a nutrient agar plate. Okay, and so remember the TS agar T and S stand for tryptic soy, and then that's the broth, the liquid version of that. And then if you add agar to make it solid, then it's an agar, and then the form that it's going to be presented in is going to be in a petri plate. So we call this media TS agar plate. If we have a TS broth tube, this would just be tryptic soy recipe. It's going to be in a broth or a liquid, and it's going to be in test tube, so it's not solid. Okay, and there's purposes for one or the other. All right, so we're going to look at the streak plate. So we have TS agar plates, or in this case, we're going to use another media. It's called Luria broth agar plate, so LB agar plate. That's this one. All right, doesn't really matter as long as our organism grows on it. Okay, so this is a drawing of our petri plate, and what we're going to do is we're going to use our flame, or if we have this incinerator here that we've used sometimes, if you look inside of it, it's red hot, and it'll sterilize the loop that way. So we're going to go in, sterilize our loop like this, stick it into there. Or if we're doing a Bunsen burner, we start up high and come down in the flame, and then go in that way, get it sterilized. And then we can go into our bacteria and get it. And then we have a bacteria on our loop, and then we just smear it. We just streak it. And what I like to do in the first quarter of the plate is just go back and forth. Go wall to wall, so you have this nice confluent growth when it grows up. Okay, then you're going to flame it again to kill all the bacteria on it, and then you're going to rotate the plate a quarter turn, and then this time you have a sterile loop with no bacteria on it. You start out here. It's a good idea to touch the loop onto the agar that's blank, because sometimes if you go in right away, it's too hot and it kills the bacteria. So we touched it, and we go in once into the previous zones and twice, and that's it. Just a couple of times in the zone, and what we've done is gone in and got a few bacteria, and we brought them out, and they trail off and are laid down on this edge of the line. Okay, and then we can then stay out from there on out and just do nice tight loops. We don't backtrack, we just keep moving forward. Okay, and then we're going to flame and kill everything on this one. And then we're ready to go for zone 3. So we're going to start out here, go in a little bit, go twice, and then we're going to stay out. And if we're only going to do 3 zones, we're just going to stay out and widen it, widen and widen it. Some teachers will do 4 zones, which means you just do this one across. It'd be like this. So here's your first zone, here's your second zone, here's your third zone, and then here's your fourth zone. But whatever your last zone, if it's the third or fourth zone, you are going to widen your area out. So the key points is never to backtrack except on the first zone, and then make sure you sterilize the loop before you go into a zone. Touch it on the surface of the auger to cool it, and then you can streak it out. And so if you then incubate this 24 hours later, there'll be a lawn, just indistinguishable bacteria. We call that a confluent growth or a lawn. And then out here, same thing, but then maybe right here you'll start to have individual colonies. So you would have a lawn right here, a lawn right here, and then you'll start having separate colonies pretty close together. But by the time you start doing that snaking, you have these colonies. And what a colony is what is a single bacterium that has been dropped off onto the surface as the loop has been dragged, and it grows and divides by binary fission into 10 million bacteria. There's probably 10 million bacteria in the size of that spot. Okay, and all those bacteria, since they divide from the original one bacterium, is called a colony, and they're genetically identical, and so they're a pure culture. So when I go in to test this, I want to find these isolated pure cultures. So when we did the simple stain last lab, I found an isolated colony, and I touched that and inoculated my slide to do a stain of it. Or I could inoculate a test tube to do a biochemical test or whatever. Okay, now this is a complete plate. Also, you'll encounter this quarter in lab half plates. And so this is a special plate that has a wall down the middle of it. And so you're going to change your technique just slightly. It's still, we'll do a 3 zone, but you're going to flame your loop, go in here, back and forth, wall to wall, and then we'll flame it again, kill everything, and then we're going to go in a couple of times, and then we'll stay out. Okay, flame, then we'll go in here, and we'll stay out, and then we'll start to snake her out. Okay, and then we can put one bacteria here, and we can do the same thing here. One zone for another bacteria, and we'll rotate it, and then we can go into this one a couple times, and stay out. And then flame it, and then we'll go into this one a couple times, and then we'll just wander around. And so we get twice the bacteria on one plate. And so it saves, if you're doing this a lot, this can save you a lot of time. At the University of Washington, where I did my microbiology degree, we had one professor there that wouldn't let her students use complete plates. They would always have to do split plates. And if it wasn't they would just take regular plates that were didn't have this wall, but then they would just draw this line down the center of the back side of the plate, and then they would just pretend that there's a wall there and they didn't cross that. And they could have 2 of there. So this is an example of a split plate. You can see here is zone 1, and then zone 2, and then I still have pretty good plate colonies. Okay, and then I just didn't do the 3rd zone. But you always, for you want to make sure you do at least the 3 zones. And if you're doing split plates, 3 zones is about all you can fit on there. If you're doing a full plate, you can fit 3 or 4 zones. When you're done, you want to label it, and you want want to label the edges of this. So this would be, you know, E. coli I would put. And it's a strain K12, so it's non-pathogenic. And then the date, you know, whatever that was, and then your initials. And then if you're doing labs, you might put lab number 7 or whatever you want, okay? And then that way you can track it, because you're going to put this in a basket or a bin of many other students' plates, and you want to be able to find your plate. And also you don't want to take somebody else's plate and then somebody is going to be upset. All right, so this is the street plate. All right, so now we'll go ahead and actually do it in part 2.